Hello and welcome to Sam Biology class. Friends, today we shall discuss about bacterial transduction. Transduction is the bacteriophage mediated gene transfer from one bacterium to another. It is a frequent mode of horizontal gene transfer in nature and is mediated by viruses. Transduction was discovered by Norton Ginder and Joshua Lederberg in Salmonella. Friends, you know that viruses are structurally simple. Often they are composed of a nucleic acid core that is protected by a protein coat known as capsid. In some viruses, a additional envelope may also be present. Viruses are unable to replicate autonomously. Instead, they infect and take control of a host cell and forcing the host to make many copies of the virus. Virulent bacteriophages replicate in their host immediately after entry. When the number of replicated phages reaches a certain number, they causes the host to lyse so they can be released and infect new host cell. This process is called as lytic cycle. On the other hand, temperate bacteriophages do not immediately kill their host cell. Instead, they insert their genome into the bacterial chromosome. This inserted viral genome is called as prophage. The host bacterium remains unharmed by this, and the prophage is replicated passively as the host genome is replicated. This relationship between these viruses and the bacterial host is called as lysogeny, and the bacteria that have been lysogenized is called as lysogen. Temperate phages remain inactive for many generations in their host. However, under certain conditions such as UV irradiation, they can be induced to switch to lytic mode of life cycle. When this occurs, the prophage is excised from the bacterial genome and then the lytic cycle proceeds. Transduction is the transfer of prokaryotic genes by viruses. Two types of bacterial transduction have been described. They are generalized transduction and specialized transduction. Generalized transduction occurs during the lytic cycle of virulent and some temperate phases. The best known generalized transducing phases are P22 in Salmonella typhimurium and P1 in E. coli. In this process, any part of the bacterial genome can be transferred. During assembly stage, when the phage genomes are packaged into capsid, some of the random fragments of the partially degraded bacterial chromosome are mistakenly packaged. As the capsid contains a limited quantity of DNA, the phage DNA is left behind. So some of the newly produced phage particles contain only the bacterial DNA. These phage particles are called as generalized transducing phage, which can inject the nucleic acid into other bacterial cells, but they cannot initiate a lytic cycle. Such phage particles only act as a carrier of genetic information from one bacterium to another. As in case of transformation, in transduction also, the transferred DNA must be incorporated into the chromosome of the recipient cell in order to preserve the transferred gene. During transfer, the DNA remains double-stranded and both the strands are incorporated into the chromosome of the recipient cell. About 70 to 90% of the transferred DNA are not integrated into the chromosome of the recipient cell. But often, these fragments become able to remain intact temporarily and are expressed. Bacterial cells that contain this non-integrated transduced DNA are called abortive transductants, and the process is abortive transductions. Such bacterial cells are partial diploids. Only in few cases, the transferred DNA is recombined into the chromosome of the recipient cell that gives rise to recombinant. This figure shows the mechanism of generalized transduction. It begins with the attachment of a transducing phase to the surface of a bacterial cell. Following attachment, phase DNA is injected into the host cytoplasm, after which bacterial chromosome is hydrolyzed into pieces. These are the DNA fragments that are formed due to hydrolysis of the bacterial chromosome. At the same time, phase DNA and proteins are also synthesized. During assembly stage, when phase genomes are packaged into capsids, some of the random bacterial DNA fragments are mistakenly packaged. So here, this is a phase particle that contains only the bacterial DNA fragment. Other phase particles contain the viral DNA. This type of phase particle, which contain only the bacterial DNA fragment, is called as generalized transducing phase, which simply act as a carrier of genetic information from one bacterium to another. The generalized transducing phase can inject the DNA into other bacterial cells. As already described, in generalized transduction, about 70 to 90% of the transferred DNA are not incorporated into the recipient cell's chromosome. So this process is quite inefficient. Only in few cases, the transferred bacterial DNA is incorporated into the recipient cell's chromosome by the process of recombination that gives rise to a stable transductant which has an altered genotype. The altered genotype of the recombinant bacterium is due to the integration of the transferred DNA into recipient cell's chromosome. This is about generalized transduction. The second type of transduction is specialized transduction, in which only specific portions of the bacterial genome are transferred by transducing phases. Specialized transduction occur due to an error in the lysogenic life cycle of phages that insert their genome into a specific site in the host chromosome. This inserted phase genome is called prophase, which remain inactive for many generations. Under certain conditions, including UV irradiation, the prophase is induced to leave the host chromosome. This induction results in rare deintegration or delysogenization, that means the prophase is excised or separated from the host chromosome. When the excision is carried out improperly, then the separated prophase carry some of the bacterial genes which are located next to the integration site. This resulting phase genome is defective as it lacks some of its gene. Because of this, the genome cannot initiate lysogenic or lytic cycle. These defective phase genomes are replicated and in the assembly stage they are packaged into capsids to form phase particles which are called specialized transducing phases. The specialized transducing phases can inject the DNA to other bacterial cells. As in case of transformation, in transduction also, the transferred DNA can be stably incorporated into the host chromosome under proper circumstances. The best known example of specialized transduction is carried out by the E. coli phase lambda. So friends, this is all about specialized transduction. For more updates, please subscribe my YouTube channel Sam Biology Class and also please share your comments. Thanks for watching.